Welcome to the cell division series. This video will focus on the cell cycle. At the end of this video, students should be able to explain the stages of the cell cycle and how the cell uses checkpoints for regulation. The cell cycle is a pattern that cells follow from growth through cellular division. Each stage is represented by a series of characteristics that every cell must undergo in preparation for replication. The cell undergoes four main stages in the cell cycle: two growth stages, a synthesis stage for replication of DNA, and the M phase, which is where the division takes place. G1 is called the gap 1 phase. In my mind, I remember G1, growth. It's a growth phase where the cell is not actively dividing. It's just making the proteins, nutrients, and necessary items for cell division to occur. During G1, the cell can go into a special suspended state called G0. This is a resting state I like to remember as the pause. Cells can exist in the paused or resting state until such time that they are ready to undergo cell division. At the end of G1, there is a checkpoint, during which time checkpoint proteins determine if the cell is ready to continue. Checkpoint proteins, or cyclins, ensure the cell is large enough and has all the proteins necessary for cell division to occur. It also double checks that the cell really does want to go into nuclear division through cell signal sensors that evaluate the extracellular and intracellular environment of the cell. When extracellular conditions are not right or not favorable for division, RB proteins enact the E2F transcript factors to halt or arrest the cell cycle until such time that the conditions are favorable again. This is the point of no return. If the cell enters the synthesis stage, it can only proceed through division or head into apoptosis, cell death. The next stage of the cell cycle is called S phase, or synthesis phase. During this time, the cell will replicate its DNA. I try to remember synthesis means synthesize, create something out of nothing. The cell's creating more. It's doubling its DNA. So now it has two identical sets of all 46 chromosomes. Think about squishing 92 chromosomes into that tiny nucleus. The G1 checkpoint cyclin is degraded by its kinase, and as the S phase comes to an end, the checkpoint protein mitotic cyclin forms, and the G2 phase begins. During the G2 or gap 2 stage, again, the cell is undergoing growth and synthesizing the necessary proteins, cytoplasm, cytosol, and organelles needed for division to occur. During G2, the cell cycle checkpoint, mitotic cyclin protein, begins checking for errors in DNA replication before continuing on toward its goal of division. If errors in replication are found, the p53 gene will be introduced to activate proteins that arrest the cell cycle and repair the DNA. Once repaired, p53 will allow the cell to continue on its mitotic journey. If the DNA is beyond repair, p53 activates programmed cell death, or apoptosis. Through cell shrinkage and DNA degradation, the cell is eliminated. Collectively, the G1, S, and G2 phases make up what is known as interphase. Upon completion of the G2, the cell will enter the M phase of the cell cycle. M phase, or mitosis, is the actual nuclear division stage. In this stage, the cell undergoes the steps necessary for nuclear contents to become doubled and then divided in half. Mitosis is subdivided into four phases: prophase, metaphase, anaphase, and telophase, or telophase. During metaphase, there's one last p53 protein checkpoint in which the cell is looking to see if the chromosomes are aligned and connected properly for the nuclear division to occur. If not, the cell cycle will be halted and apoptosis will begin. The end of mitosis, or M phase, leads into a separate stage called C, or cytokinesis. This is where the cytoplasmic division occurs, and the one mother cell becomes two daughter cells. Students should be able to describe the phases of the cell cycle. Students should be able to contrast the checkpoints that regulate the cell cycle.